Hi there, my name is Andrew Brown, and welcome to Glycolysis. This video is going to cover the entire process of glycolysis, some important enzymes and intermediates, then we'll cover how NAD+ are generated, and an important example of glycolysis in a human system. When you're finished watching this video, you should be able to answer questions like this one, which tests your knowledge of the enzymes associated with the reversible and irreversible processes in glycolysis. While this video will review glycolysis mostly as a self-contained element, it is important to remember the context. Glycolysis is really the first step in cellular respiration, the process by which sugars that had been consumed are converted into usable energy. It may also be helpful to review the entire process after watching this video, as the MCAT is more interested in how the various pieces fit together rather than having you simply memorize the discrete parts. At the most basic level, glycolysis is a process that converts glucose into two molecules of pyruvate. As we fill in the chart with the key enzymes and intermediates likely to be featured on the MCAT, keep in mind that this resource is designed to refresh your memory. We'll explore the discrete elements over the course of this entire video. So let's start by examining the enzymes involved, the first of which are hexokinase and glucokinase, depending upon which types of cells you're investigating. As glucose is taken up by its target cells, we want to make sure it won't just bind to receptors again and be transported out. That's where hexokinase and glucokinase come in. As the kinase functionality indicates, these molecules will phosphorylate glucose, effectively trapping it inside cells. This process is irreversible, which is denoted on the larger flowchart with orange arrows. To be as thorough as possible, we're going to cover some major differences before moving on to the next enzyme. First of all, as previously noted, the two kinases fulfill the same role, with hexokinase being general and glucokinase being specific to liver and some pancreatic cells. Second, the enzymes respond differently to glucose, hexokinase being most active when glucose is scarce, and glucokinase when glucose is abundant. And lastly, the enzymes are regulated differently, hexokinase by negative feedback and glucokinase in response to hormones secreted elsewhere in the body. Glucose 6-phosphate is then acted upon by a minor isomerase, which retools the 6-membered ring found in glucose into a 5-membered ring in fructose. The 5-membered ring can then be acted upon by our next enzyme, PFK1. Phosphofructokinase 1, or PFK1, which catalyzes the addition of another phosphate group to fructose 6-phosphate, is especially important to know for the MCAT because, in addition to being a rate-limiting, irreversible enzyme, it is also the control point for all of glycolysis. At the cellular level, PFK1 activity is controlled by energy reserves, denoted by either adenosine monophosphate, AMP, which signals that reserves are low, or adenosine triphosphate, ATP, and citrate, which signal that reserves are high. However, glycolysis employs an alternate signaling pathway that can indirectly activate PFK1 in response to sensed glucose availability elsewhere in the body. When fructose 2,6-bisphosphate is made by PFK2 in response to rising insulin levels, it will also activate PFK1, regardless of ATP levels already present. So, it can be seen that glycolysis imposes two controls on PFK1 to make sure that the cycle is churning as new glucose is brought in. The next enzyme in our growing chain is a simple aldolase, which breaks down the diphosphorylated hexose into two monophosphorylated trioses, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate continues along the pathway and actually will pick up another phosphate group with the help of glyceraldehyde 3-phosphate dehydrogenase to produce the high-energy intermediate 1,3-bisphosphoglycerate. Keeping in mind that glycolysis is simply an element of the larger process of cellular respiration, the hydrogen atom displaced during the formation of 1,3-bisphosphoglycerate will facilitate reduction of NAD+ to NADH, which funds ATP production in the electron transport chain, or fermentation, depending upon whether glycolysis is occurring in an aerobic or anaerobic environment. 1,3-bisphosphoglycerate's usefulness has not been exhausted there, however. With the help of another enzyme, 3-phosphoglycerate kinase, one of the high-energy phosphate groups can be used to build a molecule of ATP via substrate-level phosphorylation. In contrast to oxidative phosphorylation in the mitochondria, substrate-level phosphorylation is not oxygen-dependent, meaning it's the only method of producing ATP from glycolysis in an anaerobic environment. The resulting 3-phosphoglycerate is subjected to a mutase and an enolase, essentially repositioning the carbonyl and then pushing some electrons, to produce the pyruvate precursor phosphoenol pyruvate, or PEP. And from there, the conversion to pyruvate is as simple as irreversibly dephosphorylating PEP with pyruvate kinase. This step creates a second molecule of ATP by substrate-level phosphorylation and is stimulated by the presence of fructose 1,6-bisphosphate. That enzyme should sound familiar from our earlier step involving PFK1. Remember, the MCAT will be asking you to think critically about the scientific principles before you, so anytime an intermediate from an earlier step is used again, you should be immediately thinking, another opportunity to score some points. To go into just a little bit more depth, this type of feedback in which intermediates from earlier steps are used to activate later steps is classified as feed-forward activation. Okay, so that was a lot of information. Take a breath now as we prepare to change gears briefly by reviewing fermentation. Simply put, fermentation is a method of regenerating NAD+ from pyruvate. Because pyruvate can only be used to create greater quantities of ATP in the presence of oxygen, we won't miss it when we convert to lactate here. Also, please keep in mind that lactate is the product of fermentation in animal cells. In yeast cells, ethanol and carbon dioxide will be produced. Taken together, without NAD+, glycolysis cannot continue, making this a very important step. Hopefully that was a nice little break. Now we're going to return to the full-scale glycolysis process and identify some important intermediates. The first is dihydroxyacetone phosphate. Were you thinking that this was a dead-end product? Well, it's not. DHAP, which is converted to glycerol, ends up being instrumental in triglyceride synthesis in liver cells and adipocytes. What is more, if DHAP is not made into glycerol, an isomerase exists that can convert it into glyceraldehyde 3-phosphate and complete glycolysis. The other intermediates we should single out are 1,3-bisphosphoglycerate and phosphoenol pyruvate for their roles in ATP production. The diagram here is meant to show you that each molecule produces ATP by transferring a phosphate group to ADP. Remember also that substrate-level phosphorylation is the only mechanism of producing ATP possible in glycolysis, so don't be surprised if a question about this comes up on your MCAT. A discussion of glycolysis would not be complete without at least mentioning its role in red blood cells, which intentionally practice anaerobic respiration to preserve oxygen being carried through systemic circulation. Just think about that for a moment. If erythrocytes used up the oxygen for their own respiration, there wouldn't be any left over for metabolically active tissues. One of the intermediates we just talked about, 1,3-bisphosphoglycerate, has an additional function in red blood cells. When converted to 2,3-bisphosphoglycerate, the red blood cell can use this product to facilitate unloading of oxygen to metabolically active tissues. This brings up an important point about oxygen affinity that is best explained by looking at the hemoglobin dissociation curve. 2,3-bisphosphoglycerate, which makes oxygen unloading from hemoglobin easier, will shift the curve to the right, just like the other conditions you should recognize from biology. While hemoglobin's affinity for oxygen at the lungs is not affected, in other tissues, 2,3-bisphosphoglycerate will allow hemoglobin to release greater quantities of oxygen at a given partial pressure. This decrease in oxygen affinity is known as the Bohr effect and can be more easily remembered with this mnemonic, which reminds us that exercise will shift the hemoglobin dissociation curve to the right. Now, what about leftward shifts of the curve? For fetal hemoglobin, it is that kind of shift which allows oxygen to be transferred to the fetus across the placenta. Fortunately, 2,3-bisphosphoglycerate does not bind well to fetal hemoglobin, which keeps the fetus fully oxygenated should mom decide to get some exercise. That concludes the material this video is going to cover. Now let's apply what you've learned on some sample questions. This first one is testing your knowledge of the reversible and irreversible steps in glycolysis. The answer should be A. It might be simpler to prepare for all questions like this one by just committing the irreversible steps of glycolysis to memory and then eliminating wrong answers. Our next question is asking about the glycolysis intermediates involved in substrate-level phosphorylation of ADP. The answer to this one should be B. 1,3-bisphosphoglycerate is used to generate ATP with the help of 3-phosphoglycerate kinase. Please note here that phosphoenol pyruvate would also have been a correct answer if it had been offered. If you got those questions right, congratulations. If not, please feel free to go back and rewatch this video as many times as you need. Before we finish, let's briefly review what was covered in this video. We looked at the following enzymes and saw that hexokinase and glycolysis are part of an irreversible process that traps glucose in its target cells. Phosphofructokinase 1, part of another irreversible process, acted as a control point for the whole cycle. Glyceraldehyde 3-phosphate dehydrogenase reversibly generated the high-energy intermediate 1,3-bisphosphoglycerate. 3-phosphoglycerate kinase, part of a reversible dephosphorylation event, resulted in a molecule of ATP being formed. And pyruvate kinase, the other enzyme responsible for ATP formation in glycolysis, which happens to also be irreversible. Once glycolysis is finished, fermentation allows us to regenerate NAD+ in anaerobic conditions. We identified 3 and only 3 important intermediates and grouped them by function in triglyceride synthesis and ATP generation. We also saw a derivative of 1,3-bisphosphoglycerate, 2,3-bisphosphoglycerate, that was especially important in red blood cells, where it decreases hemoglobin's binding affinity for oxygen. Thanks very much for watching, and good luck in your continued MCAT study.